Okay, so this is the last section, and here we're going to talk about other type of projects related to cell competition and cell oscillations. So we have been talking about this process, yeah, and I told you that when we compared the cell death long time ago, we realized that the cell death was located in most of the regions in from B, C, and D, and the question was why. And I told you that they died in individual cells without showing any clear pattern. And and it seems that it's something that over the years we have been finding the same findings. We have observed the same findings in other tissues. Finally, another thing that seems to be happening in all the in different systems that we analyze during development is that the sequence of event, although the most logic one was going to be something like this. See, what we found is that there is no clear patterns, and there are many lines of evidence that or that show not only the same data, but we start finding why this could be happening, why we don't find this thing that it will be the most logic one. So I'm going to tell you what we have done with you in the last couple 3 years to try to understand this phenomena. So we have been studying not only cell competition that we discussed in the beginning of the course, but also all this topic of apical cellular oscillations. That for the people that never hear about this is imagine that you have a cell, and we start seeing that instead of getting small, they get small, then big, then small, then a little bit smaller. Yeah, that you have this. If you compare the size during at the time, you see that there are oscillations in time and size all the time. So it seems that the cells to compete, they need to display these oscillations, and we are not the first people that say that. We are just saying that this also happen in the pupal stage. So some of you have present about this paper. There is a process called the dorsal closure, and you can see that these blue cells are going to go inside. Yeah, so the mesoderm invaginations, and what they have found is that the cells, the apical area, to start getting smaller, they need to oscillate. Yeah, so instead of a gradual drop, yeah, a gradual drop like this. Oh, sorry, imagine that I made a line. What you observe, and is what they found, is that you find these pulses. Yeah, so it get bigger and start getting smaller and pulse again and pulse again. And those pulses were consistent, were happen at the same time. That there is an increase in a protein that is very important for the cell movement and the reorganization of the cytoskeleton, that is myosin. So here, one of the regulators of myosin is ROC. So you can see again pulses of myosin. Yeah, so it seems that the cells, instead of being gradual, require this oscillation. So let me. This is the lab that has been studied this a lot in the embryos of Drosophila. Let me show you one of their movies that you have seen here. In green, you will see the myosin concentration, and you will see that it starts increasing, and you will see in red the apical area. Okay, so as you can see, the cells are constantly oscillating, and then the cells go inside. But if you see, there is not a gradual changing; it's like a fluid change. And when they measured those, when they found an oscillation. Okay, let's observe it last time. If you see each one of those cells, uh, they start getting smaller, but this well, they don't get small in a gradual, and all of them get small at the same time. Uh, they start pulsing to get small. Yeah, and they start changing the shape, and many things happen. And here is when the cells go inside of the embryo. Okay, and you can see in green the increase in this myosin. So what we hypothesized like 3 years ago in the middle of the pandemic is we wanted to test if the changes were gradual in our case when the cells are being moved by the sexcom, or they also show pulses as it was described there. And also in the sexcom we have this question, but what happened also when the cells are growing? Yeah, we have the cells below the sexcom, they start increasing in size. So what do you think that is happening there? Yeah, so of course, you know that most of the data that we have found is that it seems that show that there are pulses, and we have been trying to see even if we have subgroup of cells that some of them are pulsing at the same time, and I'm going to show you the different evidence that we have found. Um, Now what I'm going to show you is that they have found that this phenomena can happen, although it happened with pulses. If you disrupt the system, it can happen gradually. Okay, so let me show you the information.
Early in Drosophila embryogenesis, a strip of epithelial cells along the ventral midline constrict their apical surfaces so that they collectively invaginate to form a ventral furrow. This apical constriction is driven by pulsatile contractions of the motor protein myosin II, as Claudia Vasquez, a student in Adam Martin's laboratory at MIT, explains.
The apical area will decrease in size as a coalescence of myosin appears in the medial domain, and then the apical area is stabilized as the myosin is remodeled, and then the cycle repeats, and that's how you get the stepwise constriction of the cell.
Myosin is activated by an upstream pathway that includes the Rho1 GTPase and its effector Rho kinase, or ROCK, which phosphorylates myosin's regulatory light chain to promote the motor's oligomerization and ATPase activity. Vasquez et al. Noticed that ROCK also showed pulses of accumulation that coincided with the coalescence of myosin II in the medioapical domains of apically constricting cells. This suggested that, rather than simply activating myosin II, ROCK might dynamically regulate the motor's pulsatile behavior.
So the idea was to test what would happen if you uncoupled the activation of myosin from its upstream regulators, namely Rho kinase, but also myosin phosphatase, which would be expected to reverse that phosphorylation and result in myosin turnover.
To uncouple myosin from its upstream regulators, Vasquez et al. Generated GFP-tagged versions of the regulatory light chain spaghetti squash, in which two key residues targeted by Rho kinase, threonine 20 and serine 21, were replaced by either phosphomimetic glutamate residues or non-phosphorylatable alanine residues. The researchers then observed the dynamics of these myosin mutants in Drosophila embryos lacking most of their endogenous spaghetti squash molecules.
So when we make a phosphomimetic mutant, which activates myosin but cannot be directly phosphorylated by ROCK, the myosin is much broader in terms of its accumulation across the apical domain and doesn't appear to undergo a coalescence event. And also with the actin, you don't see a clear coalescence in that strain. In addition, in the phosphomimetic mutants, you no longer have these really defined assembly and disassembly cycles, such that you have a much more continuous accumulation of myosin.
As a result, the apical domains of cells expressing phosphomimetic myosin mutants constricted continuously rather than in a stepwise manner.
So some of the other mutants that Claudia made were mutants that decrease myosin phosphorylation, such that you would expect to not be able to fully activate the myosin motor. And with those, what we saw is that you're still able to get a polarized accumulation of myosin, but these mutants also do not pulse. So we really think that it's a transition between low to high and then maybe back to low activity of the myosin motor, which is required to get these cycles of assembly and disassembly that are important for this ratchet-like constriction of the cell.
Okay, there is more information. Yeah, the video is a little bit longer. I don't want to overwhelm you, and so in other words, this is a field that is still growing, and we are trying to make a connection, trying to say that it's not only when the cells get small, it's not only when the cells get are dying, it's not only when the cells are invaginated. It seems that all of the cells display this type of irregular oscillations. So before some of you even start studying here, we start a project on SAXCOMs with some students from CMU that already graduate, and we told you that there were two components of the rotation. The cells grow here, while below the cells get smaller, and that you find cell death here, yeah, during this process. But there was a lot of things happening at the same time. Here I just highlight the changes in area, but their cells are also mixing and are extruding. And this is a thing that or you will study with me or you already studied it before. And what we were talking is that we have been telling you that the early stages of cancers can involve atypical changes in apical cell area. So with the crowding extrusion, we saw the cells extruding different instead of going in one direction can go to the opposite direction, that the myosin goes in the accumulating in the wrong way. So we know that these changes in apical area seems to be very important for the understanding the transition or to understand the transition from a normal to a cancerous epithelia. And if you think the not only the reduction in area that we have seen, but also the growth of tumors could be important to understand if the tumors also oscillate or don't oscillate, or this could be a landmark of cancer if we lose that oscillation. And as others have been studying, also it's very important to understand how some tissues oscillate to grow and how other tissues oscillate to get smaller. So to start this project, we said let's don't go crazy and don't study there are too many cells. So we are studying only 5 of these 5 regions. Later we said let's do only 4 because it's a lot of work. So in developmental biology course, with some of the students, we start working on trying to follow all those cells. And these were 2 of the students that work. There were 3. I couldn't find a picture of Christopher Epp, but here are all the 2 students that started this project. Jade and Lena worked in the beginning. Now she already know that she graduated and she is in medical school in Saskatchewan. And she was the one who started the project. And Malcolm was fundamental also for the project, and he not only gave a lot of feedback, and he also wrote with Jaden the paper. So here, this is what they found. If you see, we don't know if the cells oscillate or not. And it was very clear that they oscillate. In particular, this was very weird for me in the beginning that I thought that all the cells were getting smaller. What they found is that the cells sometimes oscillate, but they don't have a significant change. And they also showed that when the cells are getting bigger, they are also oscillating. And of course, as usually happens, sometimes the students leave, and we're very happy that they leave, but sometimes they graduate and the papers are not published, and unfortunately, this is what happened. And we're very happy that Eleanor is finishing this paper, and hopefully it will be done eventually soon. And next time we maybe will not send it to here, but we send it just for an undergraduate research journal, and we are still waiting. Yes, take longer. The pandemic has made this journal even slower. Okay, so ironically, a summer later, or yes, I think a summer later, we start working with our other students here with David, Eleanor, Aaron, and we actually got this paper faster published. And some of you, all of you, have participated in collecting data from this. And here there are more than 150 cells. So in the other one, there were just four group of 5. So they were 20 per movie. Here we have more than 50 per movie, and we are now working, and this is only from the top region of the COM. now our students are trying to study the bottom of the column, and we found that there were these oscillations that looked very complicated. Yeah, that we were expecting something simpler, but what we found is a lot of crazy things happening at the same time. So I'm going to very quick tell you a little bit of the stories that you have solved. And so the idea is, could we untangle all this complexity, and how do we untangle it? So some of you have worked to see if we can untangle it for using the period of oscillation. Maybe if the cells all oscillate fast in the beginning and slow later. Yeah, so there are fast oscillations in the beginning and maybe a slow oscillation there, and this is a way to understand this complexity. And we will talk about that a little bit. We will talk about the progress that we have done studying the spatial and temporal patterns. That it has been still, this is in the initial section, but shows very interesting findings. And also I'm going to show you some of the cool things that we have a study related to the cell death that they have been very interesting. Okay, but why to study collecting all these data? Yeah, why to do all these things? So many cells. Because right now, and this is a field that is growing a lot in or an approach that is growing a lot in study cancer and studying maybe all biology and all the fields, is to use computers to try to help us to analyze all this complexity. Maybe we will not be able to solve it with our own eyes, but maybe if we send all this data and use machine learning algorithms, maybe it can help us to understand better what is happening. And we are not the only one. I'm going to show you a video that show this kind of approach. Yeah, that they take, for example, slides of patients with cancer, and they digitalize the pictures. Yeah, so they put it in a computer, and the computer starts saying, okay, this shape is characteristic of cancer. They teach the software what is normal and what is going to be abnormal. Yeah, and slowly the something that it take a pathologist days, you can do it in hours, and it can get better and better, even better than that the pathologist in the beginning. So we are going to use all the data that you have collect, and we are going to train we're going to use an algorithm of machine learning to try to do the same thing and try to see if it can help us to attack all this complexity. Okay, so let me show you a video to talk about that.
There's no cancer treated without understanding what the pathology is.
The tissue slides that pathologists use to diagnose cancer contain a vast amount of information. It's very time-consuming, very fatiguing.
Every cell needs to be examined. Sometimes cancer is very difficult to recognize.
We had the idea for the microscope project about a year ago. It brings augmented reality into an existing microscope to detect cancers in real time. We're bringing AI directly to the user and meeting them where they are right now.
I think this is going to enable almost all pathologists to have access to machine learning.
Just a few weeks ago, we got it working end-to-end.
It has a camera, so it's seeing the same thing I'm seeing. Information is being fed into a computer, which is doing billions of calculations and telling me that's the tumor, that's not the tumor.
Right now, it detects breast cancer and prostate cancer, but it could also work on any other cancer type that we train models for. The knowledge that you're getting when the microscope points out cancer to you is the knowledge of many pathologists who contributed to training the machine learning model.
We had a team of pathologists that went through thousands of images, teaching the computer what cancer cells look like.
I think it has been very effective at locating areas of concern. It's like getting a second opinion.
It's going to make the doctors much quicker. They're going to make them better, which is going to overall benefit the patient.
The mission is to increase the accuracy and availability of cancer diagnosis and save people's lives. I can hardly think of anything that's more motivating.
Okay, so of course here Google is trying to sell us that they are very good. This is not. I just found that this for education purpose. This was something interesting. Yeah, it has been done by multiple groups, and it seems that it is going to be very helpful in the long term. Yeah, and it's going to revolutionize or reshape this field in the sense that it will be faster, but also the question about what we do now with the people that used to be the pathologists. So uh, I'm sure that we can that the main idea, if we do this correct, they will have more time to dedicate it to research or to dedicate it to improve the patient care instead of losing their job. But we will see. Sometimes the business are in some parts are more important than the healthcare or wellness of the experts. But yeah, so this is something that is happening and is going to continue happening in many fields. And so all the data that you have collect, we will start working in that project with Tim and with some of the students on these ideas of machine learning for several years and see what we get. So hopefully all this hard work that all of us have done, we can interpret in different ways. Now I'm going to show you some of the cool work that you have done. So I'm going to show you uh, one of the things that some of the projects that you have done. I cannot include all those things. I'm just going to include some of those. So here, this is a project that David, Aaron, Josh, and Tegan did. What they did is try to see what happened when you label these cells only and try to see the spatial and temporal patterns. And this is what I expect that I told them, well, maybe this is what is going to happen. They will be oscillating irregularly and you are going to, but all of them will start getting smaller. So we thought that maybe, as this is the region, those cells are in the region, that maybe they are under more mechanical pressure because the sexcom is moving and is moving in that area in this top region faster. Maybe we will find a more clear pattern of extrusion. Yeah, so here in this region. So I wonder you think about what is your hypothesis? Do you imagine, could you draw, do you agree with this? Yeah, with these patterns, or do you think about other patterns that you can think about it? Yeah. how the cells are getting smaller. Are they going to be maybe gradual? Are maybe oscillating? They might be oscillated in sync. They might be oscillated in, I don't know. Think about it. Stop the video. Try to think your hypothesis. And I'm going to show you what they found. So again, this is just a preliminary idea, and what they found is that there is a trend in these 3 lines of cells, yeah, and the trend is the following. Here, the cells actually don't get smaller, yeah. Here they slightly, or they have the same size or get bigger. So the trends of all those cells or getting smaller and getting bigger. And to be honest with you, I think Enari, Annika, and some of you, there were a group of students that got these cells in cells and energy, and I said, okay, I explained this wrong, and there is a mistake here, but no, they were correct, and they are opposite to what I was thinking. Those cells are actually getting bigger, yeah, slightly bigger. On the other hand, the blue ones are getting slightly smaller, and the purple ones, the ones that are further, are the ones that are getting they're getting smaller, yeah. And the question that we have right now is why this is happening and how frequent this is happening. So I don't have the answers. Please think about writing during class assignment, why do you think that this is happening, yeah. And so this is what generally happens in research, that you see patterns that they go unexpected, like unexpected, that you cannot explain, but you cannot accommodate the data to what you want. You have to believe the data more than your hypothesis. Okay, so this is pretty cool, and this is unexpected, and we need to figure out it, and maybe later we will figure out that. Another thing that we have is to ask us questions, so maybe in the future we will see if the same patterns appear here and here, or here you see something similar, and try to get a better picture of what is happening. Okay, now I'm going to show you everything related to the cell dynamics of the cells that I showed you in the beginning, that there, to my surprise, some students found that all these cells that have dots will die during development. So we have the question of when, where, what are the spatial patterns, how universal is this behavior, what is the biological meaning, and what is the implication for cancer, yeah. And some of these questions we have solved it over the years, but there is still a lot of questions. So one of this project was with Thais, Anela, Emma, and Esri. And the question that I had, that we worked together, is like, okay, what about we see all these cells here oscillating and they survive, yeah? But do you think that maybe in this yellow area you will find cells that they start getting smaller, and they disappear of the epithelium? Maybe what are the dynamics of the cells when they go into this yellow area? Yeah, so what happened? These cells, I have told you all the time about cells that survive, but what happened with the cells that die? And again, this is a lot of work that all of you did. So here, please try to propose why you think, what are the dynamics of the cells that you expect to observe in the cells when they are getting smaller. Okay, so maybe stop the video and think for 2 minutes. Okay, so these were some hypotheses, some questions that we thought. One hypothesis could be that the cells also oscillate, so there was no difference with the other cells, and the other ones, it will be that the cells gradually died. And this will have been a wonderful story and beautiful, and it's very simple, because we will find a landmark of try to understand how the cells died. That the normals will oscillate, and the ones that don't oscillate are the ones that go through cell death. However, we need to test it. Yeah. We also thought about, okay, what about the cells that they start dying? Do you think that they, when they go into those region, in that yellow region, they oscillate, or they go there and they when they go in the yellow region, they stop oscillating? Yeah. And there are many questions that we can ask. For example, we ask, do you think that when they get into this yellow region, they die very fast? Yeah. Or do you think that they can die very slowly? So they spend a lot of time, they can go back and forth between the two regions, and they end up dying. So there were about many questions about the dynamics of what happened when the cells died. And look, data. So here we found that this could be a potential yellow region. Yeah. So of the, or the behavior that of the cells when they are going to die. Yeah. So definitely all of them are oscillating. All the cells are oscillating, and when they go to that yellow region, most of them died, except few exceptions. Yeah. So it seems that the data that we have partially answer some of these questions. Yeah. Because remember, we're taking pictures every 20 minutes. Yeah. So maybe we need to zoom into take pictures every 1 minute, every 2 minutes, to try to understand this process in more detail. So look, so first question, do the cells oscillate? Yes, definitely they oscillate. All of them are oscillating. So we cannot, this is not a feature of a cell that are dying. They look very similar to the cells that are going to survive. We also asked the question of if they oscillate in that region or no, and although many of them they definitely don't oscillate, it's not because they don't oscillate. Yeah, maybe what happened is that we need to take shorter take pictures fast, like in a shorter amount of time. So instead of 20 minutes, try to see what happened every 3 minutes, every 2 minutes, and in that way we can definitely test if the cells are oscillating or not. It seems that in 20 minutes, what we have found, and I cannot show you all the data, Emma did a wonderful figure showing that most of the cells take the decision of dying in less than 20 minutes, and this is consistent to what they found in our papers. Yeah, that the cells don't, there is not a destiny. The cells, there is no, in the beginning, the body doesn't know which cells are going to die. It seems that the cells are going, they decide which cells are going to die in the last in less than 20 minutes, and the cells, when they decide which one are going to die, they just disappear. So this is why we don't observe, we get all these very rapid drops. And I already showed you this question, the answer, how long they spend here. So it seems to happen very fast, and it seems to happen in 20 minutes of less that they go into that region. Yeah, so we need to study more things. So similar to the other project, there is a lot of questions that we still have, and there is a lot of things to analyze. And the cool thing is that all the things that they found in this project are consistent with the other projects. That, for example, with the period of oscillation that Kate and Arian and our students had worked. So let me finish this telling you the last project. So I have shown you this. Sorry, I have shown you the spatial and temporal patterns. I show you the patterns of cell death. Now I'm going to finish with the periods of oscillation. That they were trying to study the pulses of oscillation. Yeah, so here you have pulses of oscillation. Yeah, so how long are those pulses? Are those pulses slow? Are those pulses fast? The longest pulses will be the ones that are the longest here in length, and the shorter one will be, for example, this one will be a short pulse that are very fast. And previous work have shown that in other systems, like in embryonic systems, these are the pulses that they have, but our process is 10 to 15 times slower. So we wanted to know if something similar happened in our system. So we create two potential hypotheses. One is that we thought maybe all this region will have the same pulses, yeah. And the other thing that we thought, like, maybe there are different subregions that have different pulses. Yeah, so we analyzed that information, yeah, and we said, like, maybe we will find this, or maybe we'll find this type of distribution of pulses, or maybe the pulses are all the same. And so far, what Kate and Ben found this semester is that it seems that, consistent to what they found in cell biology, we only have this type of pulses, yeah, and they don't have this bimodal distribution or this uniform distribution. Yeah, we have most of the pulses that we have are the 40 or 60 minutes, although we have the same type of problem that we will need to see to take pictures to answer better what happened every 2 minutes or every 6 minutes, to see if it's because we are taking pictures in the wrong time frame. They also, based on our information, the dying cells and the surviving cells show no difference, no significantly difference in the periods of oscillation, in the numbers of oscillations. This is interesting because, again, it seems that the cells decide. There is not a destiny for a cell to die or to survive. Yeah, it seems that the decision is taken very fast. Okay, so we finished all the projects that for today that I wanted to show you. There were more projects about cell shapes. They were that I didn't show you about the order disorganization. The class was getting too long. The main idea is that all the work that you have been doing in the long term, it will help us to entangle this complexity. And as you can see, the project is getting more interesting, and there is more data about how cells compete using cell oscillations at the same time. Okay, this is the last section of our class. I hope that you enjoyed, and if you have any questions, just let me know.